Hello, everybody. Welcome back. This lecture is going to focus on the material from Chapter 12, which is DNA, the molecule of heredity. So you were introduced to DNA when we were covering nucleic acids in Chapter 3, our biological molecules chapter. It is one of the four major organic molecules, right? So we covered carbohydrates, proteins, lipids, and nucleic acids. So DNA is probably the most famous of the nucleic acids. But it is also related to RNA. And we saw a little bit of ATP and NADH. Those are nucleotide related molecules. So this chapter, we're going to focus primarily on DNA. So let's get started. All right, so we're going to start with a little bit of a historical kind of walkthrough just to get us to our understanding of what DNA is and what it does at our modern level of understanding. So by the late 1800s, so the scientific community knew some things about DNA and heritable information. So they knew information was coded on these little short regions of the chromosome called genes. They knew that genes were part of these chromosome structures they could see in the microscope. And they knew that chromosomes were made out of DNA and proteins. So the DNA is organized and wrapped around proteins. And so they kind of knew that much. But the big question mark was, is it the DNA that is holding all of the code for life, or is it the protein that's holding all the code for life? And both are pretty complex. DNA is a huge molecule, but proteins are very diverse. And so there was a group of people that argued that it has to be the proteins. They have all those 20 amino acids, and they're very diverse, and they can do all the other things. So it makes sense that they would also carry the heritable information. But then they couldn't really, it didn't fit. Where the genes would be or how they would be passed on from organism to organism in the generations via the proteins. So there was kind of a race to figure out, was it the DNA, was it the proteins? So there's a couple of experiments. Your book highlights one of those with the viruses that attack bacteria. So this was the Doing Science thing right here. You can read through that. We're not going to get into that as far as content, but the outcome of these experiments was it was found that DNA was the molecule of heredity, not the proteins. So a little bit more people are getting involved. So in 1940, there was a biochemist named Chargaff that he was analyzing the DNA. They were learning that there was sugars and phosphates and these nitrogenous bases, the A's, T's, C's, and G's. And so what he did, he started looking at the ratios. How many of these different nucleotide bases are there compared to the other ones? And so from organism to organism, he was testing humans and E. coli and plants and all these different kinds of tissue samples. And he found that whenever he had a certain amount of A's, he always had a very similar amount of T's. And then the same relationship with C's and G's. And so this became known as Chargaff's rule, also known as base pairing rules, is that A's always pair up with T's and C's always pair up with G's. So these are the base pairs. And so then they got to figure out what this three-dimensional shape was. They knew there were sugar phosphates, they knew there was these bases, and now they had this new information that the bases were paired up somehow, but it took time to figure that out. So again, also in the 1940s, several scientists were involved in trying to figure out that three-dimensional structure. What does this molecule look like? So here we have a picture of Maurice Wilkins and Rosalind Franklin, and they did what was called X-ray crystallography, refraction crystallography. So it was type of an X-ray imaging. And so what you have here on the left is a picture of DNA that is like if you're looking down the barrel of it. So we know that it's a double helix, right? So if you have the double helix and you're looking straight down, this would be the outside. Let me get kind of a bright color here. So this would be the outside of the helix. This would be the central core, if you will. And these lines, the X here, those are representing how the base pairs are kind of stacked think of like a twisted ladder, right? So here's your A's, T's, C's, and G's, and if you twist it and then look down, that's the kind of picture you'll see. So it kind of gave them that the bases are pointing inwards. Some people thought they pointed outwards. It gave the size or the radius and the diameter and the circumference, how big this molecule is from that kind of looking down the barrel perspective. So that was very valuable information for kind of putting the pieces together of how all of the chemicals of the DNA come together. And then finally in 1952, Watson and Crick, James Watson and Francis Crick published their work on their structure. They took the information from Rosalind Franklin and Maurice Wilkins and Chargaff's rules, and so they kind of came up with the three-dimensional shape of DNA, and they actually won the Nobel Prize for that about 10 years later. And so here's a very famous picture of Watson and Crick. I don't know which one is which. I think that's Watson. I think he's the one with the pointing the pen, and this might be Crick down here looking up. But some cool things you can see in this picture. This is definitely a posed picture for whatever publication it was in. But a couple of things you might want to note here is in the background. Take a look. Here's their drawing. They have a drawing of that double helix showing, oops, the sugar phosphate backbone, and the base is going across this way. So I like that. Kind of if you pay attention to the background, you can see what their thoughts were. And then they had this wonderful model built to show how the nucleotide bases interact. And also what I want to point out real quick, take a look at this shape I'm highlighting right here. Right? It's a pentagon. It is a type of sugar. So this is where the ribose in the deoxyribose comes from. So we have these ribose sugars, which are part of what we call the sugar phosphate backbone. And so there's another one kind of behind his elbow here. So these sugars have these phosphate groups in between, right? So this is what's going to be bonding them together. So there's a phosphate group, sugar, phosphate group, sugar, phosphate group, sugar. So this is our alternating sugar phosphate backbone, and then the nucleotide bases are going to be these pieces in between that come off of the sugars, right? So I know that's a little bit hard to see in that picture. So let's take a look at more schematic views. So here we have the molecular structures of our actual nucleotides. So if you'll notice, they have three basic parts. So we have the phosphate group, we have the sugars, that's the pentagons I was just highlighting in the last picture, and then here are the nitrogenous bases. So when we talk about a nucleotide, it is those three parts. So we've got the phosphate group, the sugar, and the base. Okay? Now the sugar and phosphate don't change. That's going to be the same between all of the nucleotides. But what does change is the structure of the nitrogenous base. And it's called nitrogenous because it contains nitrogen. So if you'll notice, look at just the different options for the bases. So the nitrogen, right? So here's our nitrogen, 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 nitrogen containing bases. So adenine, let me get rid of some of these circles here. So adenine is a two-ringed structure, right? So you can see that it's got two carbon rings. And guanine is also a two-ringed structure. So it has some similarities, some nitrogens, but here we have an NH2 group, and here we have a double bonded oxygen. Here we have a hydrogen, and there's no hydrogen here. Here we have an NH2, and there's just hydrogen. So on the surface, they look very similar because they are two rings, but if you look closely, you'll see some of those functional groups are slightly different. And then when you come over here and take a look at the other two, we have thymine and cytosine. They are just a single ringed structure. And again, they're similar in that they're single ringed and they have nitrogen kind of in the same spot here. But we have a methyl group here where it's just a hydrogen. We have a double bonded oxygen where we have an NH2 group here. So again, some slight similarities, but also some differences. And so when we take a look at these, when we see which ones pair up, the thymines and adenines pair up. And so you're going to have a double ringed base pair up with a single ring base in that partnership. And then we have another guanine and cytosine where the double ring guanine pairs up with the single ring cytosine. And the reason why this is important, a couple reasons why, is because remember with Rosalind Franklin's picture of the diameter, these molecules, they knew the size of them. Chemistry had all of this stuff figured out. But if you had two, say like guanine and adenine trying to partner up, they would be too big and they would bulge out on the side and it wouldn't fit into that diameter of the the circular part of the DNA. If you had thymine and cytosine together, it wouldn't work either because they would be too short. They wouldn't be able to bond and reach to each other in the middle. So when you have adenine and thymine, it lines up perfectly. When you have guanine and cytosine, it lines up perfectly as far as the sizes of the molecules go and the width that they have to fit in between those sugar and phosphate backbones molecule. And another reason why these guys pair up is because of the bond that holds them together. So there's hydrogen bonds associated with the A's and the T's and the C's and the G's. So if you'll notice, that's these little guys right here. So they're illustrated as little red dots. So between thymine and adenine, there's two kind of regions where you have the partial positive and partial negative, and that's giving you the two hydrogen bonds. When we take a look at between guanine and cytosine, there are three areas between those two nucleotides. So it's kind of like this, that have partial positives and partial negatives, and that's going to give you the hydrogen bonding between the C's and G's. So even if you maybe wanted to bond an A with a C, because they'll fit, they're not going to line up properly to have those hydrogen bonds. So that's what's holding the two sides of the DNA together. And then here are your sugar, right? So here's our pentagon deoxyribose sugar, and here's our phosphate group. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. So it's an alternating sugar phosphate backbone with the bases sticking towards each other, and then the whole thing is twisted like a helix. So this is more of the molecular view right here. This is more kind of a drawing, ribbony view showing you that the blue is representing the sugar phosphate backbone, and then you can see the pairing up the bases, C's with G's and T's with A's. And then this last representation is what's called a space filling model, where they give each atom in every molecule a different color and their proportional size. So oxygens are really big, hydrogens are really small, carbons are kind of in between. So you can see it's not this loosey ribbon, it's actually a pretty compact molecule with the sugar phosphates are on the outside edge and your nitrogenous bases pointing towards the middle. All right, so then they figured it out. So we know it's DNA, it's not proteins. We know the structure, it's a double helix with the nitrogenous bases pointing inwards. But there was that kind of group of people that were like, it can't be the DNA because it only has 4 differences. Proteins have 20 different amino acids and they can fold in all of those different diverse ways. How the heck can DNA with just 4 bases code for genetic information? Well, DNA molecule is huge, but it's not the size of the molecule that gives your instructions. It's actually the sequence of those letters. So the order in which the A's, T's, C's, and G's are placed, that's the kind of secret code, if you will, the secret language of DNA that gives the instructions for how to make proteins or how to make these other cellular products that allow the cell to be a cell, these regulatory regions as well. So for example, I have written this particular sequence, A, T, T, C, G, C, A, G, G, C, A. It's just the random combination. I don't even know if that means anything. But if it did, the cell would read that completely different than this one down here, A, G, C, C, A, A, T, T, A, C. You have 4, it's like a 4-letter alphabet. You know, all the words that we can come up with are 26-letter alphabet. We can come up with just as many, if you will, with our 4-letter alphabet, the way that it gets interpreted later on the ribosome when we do protein synthesis. So even though there's only 4 letters, they can be used and they can be arranged in all of these different sequences that are going to allow us to have the diversity that we need to create life. Okay, so the next thing we're going to take a look at is how DNA replicates, how it makes a copy, because that was another important question when people were trying to figure out what's going on with the cell and passing traits on. So again, it was this question of heredity. How does information get passed from one generation to the next? So if you have a single cell and it divides to the daughter cell, it receives some of that genetic information, and they wanted to know how it did that. So we know based on the cell theory that all cells come from pre-existing cells. Cells reproduce by dividing in half. And so then they're like, how? How does that happen? I've included some pictures here of cells actively going through the process of mitosis. So here we can, each one of these little purple lines are chromosomes. And so they are visualized in the cell. And the chromosomes, we now know, these historical people now know that they are molecules of DNA that have the genes that code for information. And so now they wanted to solve the mystery of how do we get the parent cell, so say if this was the one cell, here's its nucleus, it's got all of that DNA, how do we get from that to, say, this stage where we're clearly pulling apart two sets of the chromosomes? So that's this process of DNA replication. So before a cell can divide, obviously the DNA has to make a copy of itself. So the daughter cells will get identical copies of that genetic information. And we call this process DNA replication. Make sense? So here's just a kind of a schematic picture of the parent. It would be like the parent DNA molecule in the green. What's nice about that Chargaff's rules, that base pairing rule, is because they have those special rules, A's with T's, C's with G's. If you know one side of your DNA, right? So if there's A, T, G, we know that this is going to be T, C, G, if I remember my letters correctly. So if you remove one half, we can actually use this half as our template to make that complementary half. And so that's exactly what the cell does. Because the DNA is a double helix, when you split it apart, each half serves as a template to make new daughter strands. So that's the red bands showing here, is that the new DNA is made by adding the complementary base to the parent strand, what we call the template strand. So in your book, you have this diagram. I didn't want to rewrite all the steps in the blue, so we're just going to work through the picture. So here we have the blue parental strand up here in its particular sequence, A's, T's, C's, and G's. Now, we're not teaching all the details and the enzymes and things, but just do know that enzymes catalyze all of these reactions. It's just not a random chemical process. So there's an enzyme that comes along that splits the DNA in half. There's an enzyme that comes along and reads, right? So if this is me, my big blobby enzyme, it's going to read this G and say, okay, so if it's G in the original strand, what is it going to be in the complementary strand? So it's going to add this C in there because it's reading the parent strand. So that's the stage 3. New DNA strands are synthesized with bases complementary to the parent strands. And so you have it going on both sides. So here, if we're going to see another one of those enzymes, my big red blob, if it's reading T here, then it's going to recognize that A has to go here. And if you take a look, we are identical. They're equal on both sides. And so basically we have enzymes that unzip the DNA, we have enzymes that come along and read the template strand, adding the new complementary strand, and then by the time you're done, you will have 2 identical copies of DNA, and all of that happens before the cell goes to finish its process of cell division. So this is called DNA replication. And again, it's not shown in the picture, but do realize that there are enzymes that are catalyzing all of these reactions. Okay, so there is a term that describes how this process happens, where you start with one parent strand, you unzip it, you separate it, and each parent strand serves as a template for a new daughter strand. So basically, your daughter strands are identical, with one half being old and one half being new. This is called semi-conservative replication. So semi kind of meaning half, conservative means we have half old and half new. There were some hypotheses early on that the parent strand would split, the daughter strands were made, and then the daughter strands came together and the parent strands went back together, but experiments showed that that was not the case. Semi-conservative replication is how DNA copies itself. Okay, now there are we've talked about mutations before, some accidents that can take place that can lead to natural selection and variation of population, and this is where mutations show up. The enzyme that puts in those new bases is called DNA polymerase. Polymers, right, are big long molecules made up of repeating monomers, and ACE is just an enzyme that means ending that means enzyme. So DNA polymerase polymerizes DNA. It makes it into that large molecule. And so it's got a mistake rate, if you will, about 10,000, every 10,000 bases, it might make a mistake. But we have some proofreading. We have the ability to come in and fix that mistake. And then I think the bases, the mistakes turn into 1 in maybe 1 billion instead of 10,000. So we actually don't have very many mistakes happen just in the process of DNA replication. But you could expose yourself to things in the environment that increases the likelihood that mistakes might happen. So some of those could be radiation, like UV light comes in and that could damage your DNA, increasing the likelihood of mutations. But like a mutation being the wrong base being put in to the molecule. X-rays, too much exposure to X-rays or gamma rays, those high energy wavelengths can affect your DNA and cause mutations. Exposure to chemicals, so carcinogens and cigarettes and other chemicals, processed foods, cosmetics and cleaning products, if they've been found to have carcinogenic properties, those things mean they mutate the DNA enough to change that cell cycle and cause uncontrolled cell division, which leads to cancers. And my head's kind of cutting these off, but it's viruses and bacteria. So infectious agents, you can get infection. Some viruses cause cancer. So what they do is they go in and they insert some of their own DNA, which causes mutations into your DNA, which can lead to cancers. All right. Take a look at mutations and we'll see a few examples. So again, the definition of mutation is when the nucleotide sequence of DNA is different. So you have your particular sequence. If one of those letters gets changed, that is called a mutation. It's all happening at the level of the DNA. A lot of times these mutations in the DNA will cause an outcome of the cell not working properly. Very rarely though, some of those mutations might actually result in a beneficial trait. And so that might be passed on to the population. And this is kind of how natural selection and evolution works, where you have all these random mutations. The detrimental ones get lost in the population because those organisms die out. Beneficial ones get kept in the population, kind of honing each organism to fit best in that environment. So mutations are kind of the key source of a lot of that natural selection and evolutionary selection. So there are a couple different types of mutations. We're going to take a look at the first group, which is called points mutation. A points mutation is just where it's a single base is changed, like one base is the problem. And then we'll take a look at some larger scale mutations at the chromosomal level. So there are a few types of points mutations. The most basic one is called a substitution mutation, where one base, one nucleotide is exchanged for the wrong nucleotide. So an A is accidentally put in for a T or a G or a C. And I have pictures on the next slide, we can see that. Another type of point mutation is an insertion, where the DNA polymerase accidentally puts in an extra base that just isn't going to really have a partner, but now it's inserted into that DNA. And the next time it's replicated, that extra base gets now put permanently into that DNA sequence. Or the reverse of that, as DNA is going along trying to copy, it takes one and one out, and this is called a deletion. So we have substitutions, insertions, and deletions. All of those are examples of point mutations, where it's just a single base change. So here's some pictures to represent that. So let's say on the top, this is our original DNA sequence, right? And then when it divides or it gets split apart, the DNA polymerase will come in and read, and normally will give you an exact copy. In a substitution reaction or substitution mutation, this A right here, if we follow the arrow down, accidentally gets turned into a T. So then its complementary is going to be an A instead of the T. And so that's going to throw things off. Now we haven't got to protein synthesis yet. We'll actually see how these mutations can affect the product when we get into chapter 13. All right, so here is an insertion mutation where here we have our TGA, right? So we'll go, I'm looking at the top strand here, TGA, but then when we insert it, there's our TGA, but now we have an extra T. So now it goes TTGA, and now that's going to mess up the sequence of those letters. And remember, it's the sequence of the letters that gives you the instructions for your product. That sequence is very, very important. All right, so then here is our normal CCTG, and we've lost one of our C's. So now we have CTG. So we've lost this base pair right here. And again, that's going to mess up the sequence. So if you change the sequence by a mutation, you will change the outcome or potentially change the outcome of that protein product. All right, on larger scale mutations, these are what I like to call chromosomal mutations. They're more than just a single base. They're a whole chunk or a section of the DNA. This first type is called an inversion. So they've kind of highlighted it in the darker pigments, and then the original strand is kind of the more pastel colors. But here we have mostly greens on the top. Actually, I will try to color coordinate here. So here we have greens on the top and purples on the bottom. Okay, so that would be the original sequence. And then for whatever reason, during the process of DNA replication or some other process inside of the cell, that chunk gets cut, it gets flipped. And now what we have is we have a whole bunch of all the purples on the top and the greens on the bottom. Okay, so that's the inversion, it gets flipped-flopped. And so that's going to affect the sequence, especially if this is right smack dab in the middle of a gene, you're gonna have a whole bunch of mistakes in that particular region of how that gene is expressed. This next example is called translocation. So here what they're doing, they kind of have two different sequences, kind of the pale side here on the left and the more darker pigments there on the right. And so these could be on two different chromosomes, or they could be on the same chromosome. They could be on homologous chromosomes or non-homologous chromosomes because they're all kind of coiled up together when they're hanging out in the nucleus. So there could be some effect, maybe an environmental effect or just an accident, a random problem where we kind of cut the DNA and we swap these big chunks. So if you take a look, here we have partly the pale group from this guy comes over here, and now it's attached to the darker pigmented, and then vice versa. So that's a larger scale chromosomal abnormality, which usually yields non-functional cells. So the cell might die, or if this is early on and maybe an embryological development, the whole fetus and organism might die and be miscarried because it cannot survive if it has these huge chromosomal mutations. All right, so that's kind of a short lecture, short chapter, just basics, a little bit more diving into the details of the nucleic acid DNA. So I found a cool kind of picture to end up with. So we will continue on our discussion of DNA and apply that. It's our blueprint, right? So how do we get from all this information that's stored in the DNA into the actual products the cell makes? And this is what we call transcription and translation. And we will see that in chapter 13. All right, thanks so much. I'll see you next time.